Great. Thanks, Ben. Really appreciate the time today, and again, thanks to Brian and BioPlan and the whole team that's put this together. It's really quite interesting to do the networking beforehand, have a couple of case studies, and then we'll be able to talk about it later on. So, I've been in the cell and gene therapy business for about 20 years, and so I thought it'd be good to just lay the landscape of how it's different from what we see in the monoclonal antibody space. And one of the biggest differences is that we haven't standardized on one technology uh, like we have, with CHOs and a fairly standard upstream and downstream process in the monoclonal antibody space. Viruses and viral vectors come in many shapes and sizes. Mark has just joined a company that has one type of virus and has its own unique characteristics. You still need to grow those. Use cells to grow and create the viral products and then purify it downstream. So there is an upstream and a downstream and a fill finish, but many other differences. So I was involved with a company at the very early stages where it had just been funded via preclinical studies and was looking to enter into clinical trials in the next 18 to 24 months. And it was a product called AAV, which is one of the viral vectors. It's a popular vector system that creates it, has the gene go in transiently into either direct inject or into systemically, mainly liver, CNS diseases, types of things like that, and often rare diseases. And this was an orphan drug potential in a rare disease that this product was going to be used for. The challenge was that the technology came out of academia, and the process that had been used in academia and for the early studies involved adherent processes, cell factories on the upstream side, and ultracentrifugation as part of the purification on the downstream side. There is a unique issue with AAV, with how complete the virion is during the manufacturing process, and so they call it full if it's an infectious particle, and empty if it's anywhere into the it's not complete. And so that full to empty ratio and trying to purify away the empties, ultracentrifugation is one of the process steps, but it is not, or it's difficult anyway to have it as a scalable process. So there are really a number of different ways to manufacture AAV. We could continue with this adherent process using cell factories and transient transfection, or transfer to one of the other technologies that are more in the suspension area. One is to have suspension bioreactors, again with transient transfection using plasmids to create the AAV. The other is to use baculovirus as a system, or another way is to use helper systems, either herpes virus or a wild type adenovirus to provide some of that information that you would normally get from the different plasmids. When the company looked at the potential commercial need, it drove almost immediately towards, we need to be at the maximum size of reactor that we can to be able to meet what the potential need is based on the productivity that they were aware of at that time. So this wasn't involved with any productivity changes. And I will say that, again, comparing to monoclonal antibodies in the gene therapy space, there's no question of whether or not you're going to use disposable reactor systems. So it is the most widely preferred process. So is to use subs and disposable equipment. And just one part of that is that that these could be considered infectious products. And it's much easier, you don't have the clean validation and the things that go along with that. So how do you scale? You could go into suspension with transient transfection all the way up to the maximum scale, which is 2,000 liters. But the phase 1-2 trials, it would not require even close to that. This was aspirational. But the adherent processes to get any scale on that, you really can't continue to use cell factories. It would be enough for the phase 1-2 trial, but it was not going to meet the needs for a phase 3 pivotal, and clearly not on the commercial side, again, based on how it was picked up. And in the rare disease space, if there are only 1 or 2 or 3 companies that are going after that, it's, you can capture quite a bit of that market if you're successful. So it's a little different again in the market size. Another way that you can scale is to remain an adherent process, but then go into a fixed bed bioreactor. And so there are a couple of companies that use fixed bed bioreactors, and there are AAV products on the market right now that have used fixed bed bioreactors, have made the decision to go into commercial with that process,
right.
And not change, just stay with that process from, I think it was, again, early phase 3 pivotal, certainly into commercial. So those were the different areas that people were looking at, and kind of the continued challenge that we all face, but may be exacerbated in the gene therapy or rare disease space, is that the trials were usually condensed. So you were seeing phase 1-2s where you're having, you're treating tens of people rather than hundreds. And, the intent now is to go from a phase 1-2 straight into a phase 3 pivotal. And that's not going to allow conversion to, or often won't allow conversion to a new process. So many challenges that were going on, let alone that the investors that had just invested in this company wanted to start the clinical trial as soon as possible. And their preference, at least in the beginning, was to stay with the academic process, get it good enough, and get into the clinical trial to see if it would work. And so a number of us were looked at all of these different options, obviously, including staying with the academic process, and went through and did a complete study as to the merits of each, and came up with a decision to go to suspension at 50 liter, and take out the ultracentrifugation, to use some alternative steps to try to help with that full to empty. The BIA system was implemented. And also, what I didn't talk about was that the timelines to get into the CDMOs was, for a new company, very, very challenging. It was, 18 months, 24 months. And again, as a newly financed company, those timelines were very difficult to hit. So there was a discussion to also look at building own manufacturing facility, in addition to looking at CDMOs. So the decision was really to look at 50 liters suspension, transient transfection, with a downstream process that had moved away from this ultracentrifugation. There was a CMO that was selected that had a slightly stronger and better timeline, where we could get product in 9 months at the 50 liter scale. And the selection criteria for the CMOs involved the ability to go up to 500 liters from that 50 liter. So there were bids for 50 liter and a bid for 500 liter. The facility design on the build versus buy side was also designed, but only to 500 liters. The decision was made not to go up to the maximum scale, which was 2,000 liters. And that was primarily due to just the large distance of going from the initial academic process all the way up to 2,000 liters. And so it was felt that 500 liter and multiple batches of 500 liters was less risky and easier to get to, frankly, from the 50 liter scale than the 500 scale. So the lessons that came out of this was that you can't just look at the studies that say what the demand supply imbalance is and what is the fastest way to get into the clinic. That it really made more sense to take a little bit more time, and it was probably a 9-month delay on the clinic on the initial timelines to switch over to a reasonable sized phase 1/2 that was in suspension rather than adherent, which gave the best process and the best chance to be able to scale up to this 500 liter. Other companies have decided to stay with the adherent and moved into fixed bed, and I think that's a viable alternative and has been employed. But for this product and the opportunity that had it on the back end, I think it was it ended up being a better solution to go to suspension. The other part that was surprising was looking at the CMO landscape, because there are all of these different ways to make AAV, that you can't assume that all of these manufacturers have expertise in all of the different areas. So we found some CDMOs that were AAV manufacturers, but they were pure baculovirus players, and that was not a good option for us. Other companies claim 2,000 liter capability, but when we really got into finding out, it was hundreds of liters was the actual experience that these companies had. But it was a good solution. Ended up product came from the CMO on time at a moderate scale, at the 50 liter scale. I think it's now in the COVID area, and the timeline of getting to the 500 is TBD. But I think it was a good solution, at least for the initial phases.
Yeah, beautiful. So thank you very much, Dave. I did, as part of the networking, get a question earlier today for you, and that's, on the topic of CMOs, still play a role even if the sponsor builds their own facility?
Whoops, Ben, maybe it was just me, but I lost you there for a second on the question.
Oh, sorry. Can you hear me now?
There we go. Yeah.
Yeah. So wondering if CDMOs still play a role even if the sponsor builds their own facility.
Yeah. I think it's, and this is a personal opinion, but my belief is that it's very short-sighted to lose CMO relationships when the decision is made to build your own facility. Not only is it a de-risk, if you manufacture on your own, you lose the opportunity that if 5 years hence there's a strategic decision or there's a product failure, and all of a sudden a facility that you have built for multiple compounds is a white albatross. And so I think that many companies go CMO only, and you will see a number of companies in the gene therapy space where they will announce that they are building a facility, but in that same announcement they will make sure to say that they intend to keep their network on the CDMO active. It's hard to keep 2 or 3 different places active when you have your own manufacturing facility. The dynamics are obviously going to drive towards fully utilizing your own facility. So usually having your own facility and one trusted service supplier makes a lot of sense. And again, in gene therapy, it's a very, very rare time when you can become an internal expert in plasmids, in viruses, in cell therapy, right? Allogeneic, autologous, iPSCs, gene editing. These are all different technologies, and you're going to need the service network in some way or form. This company had a 25,000 square foot virus facility, but still kept the network alive.
Right. Beautiful. There's a question in the general chat that I'll let you, if you don't mind, answer offline. And
okay.
Thank you very much, Dave, and we'll see you again soon for your next panel talk.
You got it. Thanks.
Excellent. Thank you.